Hello, my dear students and all my friends. Welcome back to Immunogenetics short lecture. Today, we shall be discussing a very intricate and interesting topic in a simpler way, that is VDJ recombination that landed for Nobel Prize in 1987 by Tonigawa. So, our topic is VDJ recombination. Now, think of how many ways you can arrange a Rubik's Cube. Trillions of permutations and combination is possible. Similarly, think of a human gene. Try to understand from the basic level. We have 10 to the power 5 or 1 lakh gene, close to that. But this particular number of genes can recombine in such a way that it can form 10 to the power 15, more than trillion times. Trillion is 10 to the power 9. So what are the methodology and way to produce this diversity of a particular antibody for a particular antigen? So this specificity with diversity is caused due to a certain methodology in genetics known as a VDJ recombination. V for variable, D for diversity, J for joining. So we'll understand this step by step. Think of it in a bone marrow. In the bone marrow, there are progenitor cells and B lymphocyte that is nurtured. And finally, this will be converted to plasma cell. I have already depicted this picture, so it will save your time. So this plasma cell will produce thousands of antibody. When it is active, approximately research data says 2000 antibody per second. So it's a 1000 of antibody they are bombarding. Now try to understand what happens in the progenitor cell so that they can recombine in such a way and produce this trillions of antibody that is helping us to fight with the different types of bacteria, virus. It's a very important topic. Tonigawa, you remember this name, he got Nobel Prize for the antibody diversity discovering of this concept. Now you see, think of this B cell, it has a DNA. In the DNA region, we have a V, D and J segments. This is the part of the DNA. Okay, this is the segment of DNA, initial of the leader sequences, and say this is your heavy chain. First, you know the structure of the antibody. You know the class 12 we have read about. So in the antibody structure is the H2L2 configuration, 2 heavy and 2 light chain. So in the orange color, I have depicted the heavy chains with the VCC constant region, the variable regions, and the same way in the light chain you can see here the variables and the constant, this part. So this light chain will be translated later on. First we talk about the heavy chain. Now you think there are suppose 51 V segments. In the V region you have a different number of segments. It says if we in the V region have a 51 segment, in the D you have the 27 segments, and in the J you have a 6. And you know the permutation and combination rule, how many ways we can arrange. So I can take one from here and one from here and two from here, say for example 2 from here. So in this way you can recombine the VDJ gene segment. Now I can take also here 3 out of it, I can take 1. So in this way, how many times we can arrange it? So multiply 51 to 27 into 6, it comes around 8200. Now after somatic hypermutation, you can produce trillion number of antibody, the VDJ segment. So you can have the diversity in the antibody. This is the procedure. Now try to understand step by step. After each and every V and D segment, you have an intron region that will be definitely spliced during the splicing reaction. So if you consider first formation of the heavy chain here in the picture, so first is D and J recombination. Please note that in the light chain, we don't have any D segment. We don't have this D in the light segment. Okay, and you must know this light chain having two variation, kappa light chain and lambda light chain. I think you can see here, and all of these genes are coded from the somatic genes. So here you see chromosome number 2 code for kappa and chromosome number 22 for the lambda. Remember chromosome number 14, that is also somatic, not a sex chromosome, code for this heavy chain. Now what will happen? This D and G segment first recombine. Okay, and the constant region is as it is. A C mu, it code for a immunoglobulin M. So that will be the different segment. You see here C segment that is coming from the constant region. So now DJ recombination will happen, and the next step, this V, D, G, J and C particularly, these three will assemble. Now this way they will form the VDJ recombination will form this heavy segment with a but different types of combination. I have given orange, it could be a 1,000 different types of combination. Okay, now one more thing, you see this one, how this joining happen. Okay, think of a thread. So this is a sequence, you have a V segment here, D segment and J. Now there is a particular signal called RSS signal. I hope you can see, you take a screenshot of it so that it will help you. The RSS signal, it is of two types. What is RSS? I think you can see this. Extremely sorry, my phone actually slipped away, fell down. Okay, again we come back. So in the VDJ segment you see here, so there is a particular signal sequence called as a recombination signal sequence. The signals sequence are very important for the identification, and you know there are two types of signal sequence. This is very important, 1 turn and 2 turn. 1 turn means the gap between this sequence is 23 base pair, and for the 2 turn sequence the gap is only 12 base pair. So what will happen, they will make a structure like this, and there is a one enzyme that is very important, recombinase enzyme. This recombinase enzyme now it is coming and sitting, and it will make the cut. Okay, recombinase is like it's a cutting the RSS, it bind with the RSS and making the cut. Now again this will be joined, because you have to, in recombination you know the segment has to be cut and rejoined. Here NHEG, non-homologous end joining, will be taking place with the help of many different types of protein. Two important proteins I have written, Q70 and Q90, Q70 and Q80, sorry. Okay, now so this way you see the joint will be produced by non-homologous end joining. So this way you can get a completely new recombinants, the DNA that will again code for a RNA that will be coding for a protein. Okay, now question is that who triggers this formation of DJ proliferation in the heavy chain? In the heavy chain, you know, first DJ will recombine, then VDJ. You know that in the bone marrow, the signals and different chemicals, chemokines will trigger the synthesis of the antibodies from the plasma cell. First you see, say this is your B cell and this is your stromal cell in the bone marrow. Now these two cells interact with the KIT and SCF. These are two kind of a molecular ligand, so they will interact, and chemokines, very important, is CXCL12. CXCL12, this is a chemical signal that will trigger this B cell to proliferate and produce the antibody through DJ recombination. These are the chemicals involved in it. So this is the basic outline that we must know. And you know, for the light chain synthesis, only VJ recombination will take place because D segment is not present. Okay, so here you see D segment is absent, so only VJ recombination with the C chain. So through VJ and the C, this will help in the formation of light chain, maybe the kappa and lambda. This is the process of VDJ recombination. Through that you can produce 3 into 10 to the power 11, so more than trillion types of antibody, and the diversity is caused due to this recombination. Okay, thank you so much. I think it will help you to understand. Thank you for listening. Please subscribe if you have not, because it is free.